Hey everyone, Doctor here from Special Biomed. Welcome to this deep dive into mastering histological tissue selection. We all know accurate diagnosis starts with a strong foundation, and that's exactly what we're building today. Over the next few minutes, we'll break down the essential principles that guarantee your histological procedures lead to the most reliable results. Whether you're a seasoned pathologist or just starting your journey, understanding these principles is key for success in the world of diagnostics. Let's get started. Alright, let's jump right into our first principle: tissue representation. This is a fundamental concept in pathology and diagnostic medicine. This is all about making sure the tissue sample you select truly reflects the patient's condition. Think of it like choosing the perfect scene from a movie. It needs to tell the whole story. Just as a movie scene captures the essence of the plot, a well-chosen tissue sample captures the essence of the patient's health issue. We want to see the problem area, but also some of the surrounding normal tissue for comparison. This dual view helps us understand the extent and nature of the disease. By comparing the diseased tissue with the healthy tissue, we can identify subtle changes and patterns that might be missed if we only looked at the affected area. This gives us a complete picture of what's happening at a microscopic level. The details revealed under the microscope can be crucial for an accurate diagnosis. It's like having a magnifying glass that shows every tiny detail, helping us to see the full scope of the issue. Imagine trying to diagnose a skin rash by only looking at a tiny patch. You'd miss crucial context. The same principle applies to tissue samples. Without a comprehensive view, we might overlook important information that could affect the diagnosis and treatment plan. So remember, representative sampling is key. It's not just about taking any sample, it's about taking the right sample. This ensures that we have all the necessary information to make an informed diagnosis. Proper tissue representation can make a significant difference in patient outcomes, guiding effective treatment strategies and improving overall care. Now let's talk tissue types. Each tissue in our body is unique, kind of like different fabrics. You wouldn't wash silk the same way you wash denim, right? Same goes for tissues. A delicate brain tissue needs a gentler approach than, say, a tough bone sample. That's why understanding the specific characteristics of each tissue type is crucial. We need to adjust our techniques accordingly, ensuring optimal preservation of the tissue structure. This might involve different processing methods, fixation times, or even staining protocols. Remember, treating each tissue with the respect it deserves is key for accurate analysis. Now, let's discuss size and thickness. In histology, size definitely matters. The dimensions of the tissue sample can significantly impact the quality of the results we obtain. If a sample is too thick, our fixatives and stains won't penetrate effectively and will end up with a blurry, uninterpretable mess. This is because the chemicals used in the staining process can't reach the inner layers of the tissue, leaving them unstained and difficult to analyze. Think of it like trying to read a book with pages stuck together. You can't make out the words clearly, and the story becomes impossible to follow. On the other hand, if the sample is too small, we might miss crucial details. Important structures or abnormalities could be absent from the sample, leading to incomplete or inaccurate diagnoses. It's about finding that Goldilocks zone, not too big, not too small, but just right for optimal processing and visualization. This balance ensures that the fixatives and stains can work effectively, and that the tissue's architecture is preserved for accurate examination. The ideal size can vary depending on the tissue type and the structures we're interested in examining. For example, a liver biopsy might require a different thickness compared to a skin biopsy. Each type of tissue has its own unique requirements to ensure the best possible results. Precision in cutting and preparing these samples is crucial. Using the right tools and techniques, histologists can achieve the perfect thickness, ensuring that every detail is captured and nothing is overlooked. When done correctly, the results are clear, detailed slides that provide invaluable information for diagnosis and research. This meticulous process is what allows pathologists and researchers to understand diseases at a microscopic level and develop effective treatments. Chapter 4: Tissue Integrity. Let's move on to tissue integrity, which is all about handling our samples with care. We want to avoid causing any damage, like tears, compressions, or shrinkage, which can distort the tissue architecture. Imagine trying to put together a puzzle with bent or torn pieces. It just won't work. That's why it's essential to use proper handling techniques during biopsy, transport, and processing. Think of yourselves as guardians of these tiny universes, ensuring they arrive at the microscope slide in pristine condition. Chapter 5. Fixation time. Now, let's talk about fixation time. Fixation is a critical step in histology and pathology, ensuring that tissue samples are preserved in their most natural state for further examination. Fixation is like hitting the pause button on tissue degradation. It halts the natural processes that would otherwise break down the tissue, allowing scientists to study it as if it was still part of a living organism. It preserves the tissue in a lifelike state, preventing it from breaking down. This is essential for accurate diagnosis and research, as any degradation can lead to misleading results. But here's the catch. Different tissues have different fixation needs. The type of tissue, its size, and its cellular composition all play a role in determining the optimal fixation process. Some tissues are like delicate flowers, wilting quickly without proper care. These tissues require gentle handling and precise timing to ensure they are preserved correctly. Others are more robust, but still need the right treatment to maintain their structure. Even the sturdiest tissues can suffer from improper fixation, leading to structural changes that can affect analysis. Using the appropriate fixative and optimizing the fixation time is crucial. The choice of fixative and the duration of fixation must be tailored to the specific needs of the tissue to achieve the best results. Too short, and the tissue might degrade. This can result in loss of cellular detail and make it difficult to obtain accurate information from the sample. Too long, and we risk altering its structure, leading to inaccurate interpretations. Overfixation can cause the tissue to become hard and brittle, making it challenging to cut and stain for microscopic examination. Therefore, meticulous documentation and adherence to protocols are essential. By carefully recording the fixation process, scientists can ensure reproducibility and reliability in their findings. In summary, fixation time is a delicate balance that requires expertise and precision. It is a foundational step that underpins the accuracy and reliability of histological and pathological studies. Chapter 6. Anatomical location. Now, let's talk about anatomical location. This principle highlights the importance of knowing where our tissue sample comes from within the body. It's like reading a map. We need to know our starting point to understand the journey. Different parts of the body have unique histological features. For instance, a skin biopsy from the arm will look very different from one taken from the scalp. So, when we receive a sample, we need to know its precise anatomical location to make accurate diagnoses and interpretations. Chapter 7. Sampling method. Let's move on to sampling methods. The way we collect our tissue samples can significantly impact their quality. It's like choosing the right tool for the job. A hammer is great for nails, but not so much for screws. Similarly, different clinical scenarios call for different biopsy techniques. We have fine needle aspirations for accessing deep-seated lesions, punch biopsies for sampling skin lesions, and endoscopic biopsies for reaching internal organs. Choosing the appropriate method ensures we obtain the best possible sample for diagnosis. Now, let's discuss post-sampling handling. Once we've obtained our precious tissue sample, we need to treat it with the utmost care. Think of it like transporting a delicate piece of art. We want to make sure it arrives at its destination in perfect condition. This means placing the tissue in the correct fixative immediately after collection to prevent degradation. Any delay can compromise the tissue's integrity, leading to artifacts and making diagnosis difficult. Let's talk about orientation. Imagine slicing a loaf of bread. The way you cut it determines what you see inside. Cut it straight and you see all the layers. Cut it at an angle and you get a different perspective. The same applies to tissue samples. Proper orientation during embedding and sectioning is crucial for revealing the necessary anatomical structures. We want to make sure we're looking at the tissue from the best possible angle to make accurate diagnoses. Finally, let's emphasize the importance of communication. Histology isn't a solo mission, it's a team effort. Pathologists and clinicians need to work hand in hand, communicating effectively to ensure the tissue being selected is relevant to the clinical question at hand. It's like solving a mystery. The more clues we share, the better our chances of cracking the case. Clear communication ensures we're all on the same page, maximizing the chances of a timely and accurate diagnosis. So there you have it, the key principles for mastering histological tissue selection. By following these principles, you can ensure the success of your histological procedures and the reliability of your findings. Remember, accurate diagnosis starts with a solid foundation, and that foundation is built on these fundamental principles. Thanks for joining me today, and don't forget to subscribe, like and share for more informative content from Special Biomed.